Hello friends, warm welcome in this video, that is the enzymes without cofactors or mechanism of reactions catalyzed by enzymes without cofactors. Friends, enzymes which operate without cofactors tend to be relatively small and have relatively straightforward reaction mechanism. For these reasons, such enzymes were among the first to be investigated in detail. These are some examples which play a significant part in the development of current knowledge of the enzymes, which includes chymotrypsin, ribonuclease A, lysozyme, triphosphorus and other enzymes like these. So we will learn mechanism of reactions catalyzed by all these enzymes one by one. The very first enzyme is chymotrypsin. The serine protease chymotrypsin, a monomeric enzyme, is produced in an inactive form by the mammalian pancreas, a zymogen form called as chymotrypsinogen. This is the X-ray crystallographic structure of chymotrypsin, which reveals that only about 40% primary structure of this enzyme is similar to trypsin and elastase, the enzymes of a group serine proteases. The pH optimum for chymotrypsin is about 8. Chymotrypsin is a endopeptidase hydrolyzing peptide bond in the middle of peptide chain with different specificities. Chymotrypsin cleaves peptide on the C-terminus side of hydrophobic residues like phenylalanine, tyrosine and tryptophan as shown in this figure. Chymotrypsin is synthesized in the pancreas as the zymogen chymotrypsinogen or pre-chymotrypsin. This is a single polypeptide of 245 residues containing 5 intrachain disulfide bridges. On passing into the intestine, where proteolytic enzymes are required to digest dietary proteins, chymotrypsin is attacked by trypsin. This breaks the peptide bond between arginine 15 and isoleucine 16, producing pi-chymotrypsin. Already the molecules has a full enzyme activity, but further change then takes place. A dipeptide is removed from position number 14 and 15 by the action of another molecule of pi-chymotrypsin, producing delta chymotrypsin. And further chymotrypsin digestion removes a dipeptide from position 147 and 148 to give the final product alpha chymotrypsin, which is a active enzyme. Chymotrypsin contains 3 polypeptide chains linked by disulfide bridge, so it is not strictly a monomeric enzyme, but the sequential numbering systems of original chymotrypsin molecule is usually maintained. Chymotrypsin catalyzes the cleavage of peptide bond at the carboxyl side of aromatic amino acid, that is phenylalanine, tyrosine and tryptophan residues. It will also hydrolyze variety of amides and esters, and these artificial substrates have been used to investigate the enzyme in great detail. Chymotrypsin catalyzes hydrolysis of an ester proceed via formation of an acyl enzyme. This is also true for the hydrolysis of amides, and we can write the reaction in general terms as follows. E plus RCOY, that is the ester or amide, gives ERCOY, which yields YH, and acyl enzyme ECOR, which on hydrolysis gives ERCOH. Substrate could bind to serine 195, and also that histidine 57 was involved in reaction mechanism. The X-ray diffraction studies revealed the presence of hydrophobic binding pocket for aromatic side chains at the active site. Such studies also showed that the aspartate 102 buried in the interior of the molecule could be closely linked to the action of histidine 57 and serine 195, possibly setting up what was termed a charge release system, with aspartate 102 removing a proton from histidine 57, thus making it easier for the latter to remove a proton from serine 195 during the course of the reaction. Detailed hydrogen, carbon and nitrogen NMR studies showed that histidine 57 has a relatively normal pK value near 7, at least in the free enzyme, casting doubt on the charge relay theory. However, investigations using site-directed mutagenesis have shown that the 3 amino acids, if not setting up a charge relay system, as such nevertheless act in a concerted fashion, perhaps explaining why several enzymes have a catalytic pride of this type at the active site. An outline reaction mechanism for the hydrolysis of an ester or amide by chymotrypsin is as follows. Histidine 57 acts as a base catalyst to enable the oxygen of serine 195 make a nucleophilic attack on the carboxyl group of the enzyme bound substrate. An unstable tetrahedral intermediate is formed whose negatively charged oxygen atom may be stabilized by hydrogen binding with a backbone NH of a glycine 193. Similarly, the positive charge on the imidazole ring of histidine 57 is stabilized by electrostatic interaction with aspartate 102. The imidazole group of histidine 57 then acts as an acid catalyst to facilitate the liberation of the first product YH, leaving behind the acyl enzyme. The second stage of the reaction, like that, is initiated by nucleophilic attack, this time by water. The liberation of the product RCO is again assisted by acid catalysis. The same mechanism is thought to occur for peptide substrates. The second enzyme is ribonuclease. Bovine pancreatic ribonuclease is the first enzyme studied in great detail by enzymologists and was the first enzyme to have complete amino acid sequence determined. This was achieved by Stanford Morey and William H. Stein, for which they have awarded Nobel Prize. Ribonuclease catalyzes the cleavage of the phosphodiester backbone of ribonucleic acid by the reaction involving transfer of phosphate group from the 5th prime position of one nucleotide to the 3rd prime position of the next nucleotide in the chain. Histidine 12 and histidine 119 both contribute to catalytic process, one possibly acting as a proton donor and the other as a proton acceptor. Similar investigations have suggested that lysine 41 is also involved. The three-dimensional structure of the enzyme as revealed by X-ray diffraction studies shows these three amino acids sited close together near a substrate binding cleft in the molecule. Other noteworthy features of the structures are the presence of 3 alpha helices and 3 beta hairpins. In 1962, Brian Rabin and co-workers suggested the following mechanism for the reaction. Histidine 12 removes a proton from the substrate, producing a nucleophilic oxygen atom which attacks the phosphate. Histidine 119 donates a proton to the complex and the first product RO is liberated. The rest of the reaction is the reverse of the first part, with water replacing the ROH. This mechanism is consistent with the evidence now available. It is likely that lysine 41 helps to stabilize the complex. It is positively charged amino group forming an electrostatic interaction with a negative charge of the phosphate. The third enzyme is lysozyme. Lysozyme catalyzes the hydrolysis of the polysaccharide consisting of alternating N-acetyl glucosamine, that is NAG, and N-acetylmuramic acid, that is NAM, units, a component of peptidoglycan, which gives shape and rigidity to bacterial cell wall. The lysozyme which has been most studied is that from hen egg white, the first enzyme to have complete three-dimensional structure determined by the use of X-ray crystallography. These investigations and subsequent ones reveal that the enzyme has large cleft which can accommodate 6 of the substrate amino sugar units. This may be represented diagrammatically as shown in this figure. Each of the 6 amino sugar is bound to the enzyme by a hydrogen bond. Example, those in site A and B to aspartate 101, that in the site B to tryptophan 62 and tryptophan 63, and that in site F to asparagine 37. NAM, because of its large lactyl side chain, cannot bind to sites A, C or E where there is a restricted space. The hydrolysis takes place between the units in site D and E and therefore must always involve the beta-1,4 glycosidic link between C1, the reducing end of NAM, and C4 of NAG. The amino acids near this bond are glutamate 35 and aspartate 52. In 1967, David Phillips and Charles Vernon Both suggested the following mechanism for the reaction. The substrate binds to the enzyme and hexose ring D is distorted to less stable half-chair conformation, which facilitates the formation of a carbonium ion also having a half-chair conformation. Glutamate 35, which is in a non-polar environment and thus protonated at pS 5, can act as a general acid catalyst and donate its proton to oxygen of the glycosidic bond, causing the bond to break. Aspartate 52 is in a more polar environment than glutamate 35 and is negatively charged at pH 5. Therefore, according to this mechanism, it can stabilize the carbonium ion by electrostatic interaction. The first product leaves and the reaction is completed by nucleophilic attack on the carbonium ion by water. However, Stephen Withers and colleagues in 2001 by a variety of techniques including the replacement of the catalytic carboxyl group by an amide and the use of electrospray ionization mass spectrometry demonstrated that this scheme was only partially correct. Although it seems that ring D of the substrate is indeed distorted on binding, the reaction proceeds via the covalent attachment of this ring to aspartate 52, which thus has an important role as a nucleophile rather than simply stabilizing a charged transition state. The reaction is presumably completed by the hydrolysis of linkage between the ring D and aspartate 52. This mechanism involving a covalent catalysis is also common to other glycosides. The next enzyme is triose phosphate isomerase. Muscle triose phosphate isomerase TIM or TPI is a dimeric enzyme which catalyzes the interconversion of a glyceraldehyde 3-phosphate and dihydroxyacetone phosphate DHAP. X-ray crystallography has revealed that each subunit has an inner cylinder of 8 strands of a parallel beta-plated sheets linked by predominantly helical regions which form an outer cylinder. This arrangement, also found in some other enzymes, is termed as alpha-beta barrels or TIM barrels. Each of the two identical binding sites of a TIM contains lysine 13, histidine 95, glutamate 165 and glycine 232 from one subunit and phenylalanine 74 from the other. The reaction mechanism apparently involves glutamate 165 acting as a base catalyst and histidine 95 as an acid one. So in this way the reactions without cofactor proceeds. Thank you.